morning all of you. I am Pooja Nagotra from Paramedical Department, Assistant Professor, and my topic is ABO Blood Group System. ABO Blood Group System is the most important blood group system in human blood transfusion. A blood type is a classification of blood based on the presence or absence of intermediate antigenic substance on the surface of RBCs. These antigen may be protein, glycoprotein and glycolipid depending on the blood group system, and some antigen are also present on the surface of other types of cells of various tissue. There are nearly 300 blood group system discovered. Of these, ABO, blood and RH are major important. Discovery of blood group system. The discovery of the ABO blood group system by Carl Landsteiner in 1900 at the University of Vienna. Landsteiner work laid the foundation of understanding the Landsteiner law, which states then the serum of an individual when the antigen is present on his red cells. In ABO blood group system, agglutination reaction takes place between the surface protein and they are found on the red blood cell surface. The surface protein called antigen or agglutinogen. The antigens are mainly glycoprotein, which are of the two types name of self antigens and foreign antigen. If the antigen is foreign or will introduce the immune system response, after which of the WBC are come into action to recognize the antigen. Then the WBC start making antibody against the particular antigen, and antibodies are the molecules that work against the antigen and also called agglutination that float in the blood plasma. Landsteiner law. Landsteiner law, if an agglutination or agglutinogen is present on red blood cells membrane, the corresponding agglutinogen must be absent in the plasma. If an agglutinogen is absent on the red blood cells membrane, then the corresponding agglutinin must be present in the plasma. The ABO blood group system in type of AB and O. The phenotype on it is used to identify the type of surface antigen and antibodies present in the donor blood, and it agglutinations occur in the RBCs, then the corresponding antibody must be absent in the blood plasma. The antigen absent in the RBC membrane, then the corresponding antibodies must be present in the blood plasma. Individual above 3 to 6 months have been naturally according to include the which are the without and antigen stimulating the materials antigen stimulating the antibodies when the IgM and the ABO blood group system is the proper. Sometimes they had the corresponding antibody in the and ABO blood group method. First, scrub the middle finger with the cotton swab, the prick the finger by the sterilized needle or lancet. After that, place 3 drops on a clean glass slide, then the anti-sera in a sequence of anti-A in first drop and anti-B in second drop and anti-D to the third drop. Mix the blood with the anti-sera by using applicator stick. Tilt the slide back and forth and observe for agglutination. Interpretation of result based on agglutination reaction. If the agglutination occur in the RBCs to which anti-sera A is added, then the blood group is A. When agglutination occur in the RBCs to which an anti-B is added, then the blood group is AB. If agglutination occur in the RBCs to which both anti-A and anti-B are added, then the blood group is AB. When there is no agglutination occur in the RBCs, then the blood group is O. Next. ABO blood group system is the fundamental blood banking and the transfusion practice. Knowledge of ABO compatibility is the crucial of prevent transfusion reaction and ensure the patient safety. And ABO transfusion matching the ABO blood group type between donors and recipient is the essential for the successful organ transplantation, and incompatibility can be lead to rejection and complication post transplant. ABO and pregnancy. The ABO system plays a role in pregnancy, and maternal fetal incompatibility between material and fetal blood types can lead to a hemolytic disease of the newborn. ABO blood group disease and immune disease. Research suggests to a potential link between ABO blood group type and risk of autoimmune disease. Understanding these are association may be provide insight the disease and mechanism and the treatment of strategies. Challenges of limitation. Deep side the significance, the ABO blood group system poses challenges in cross matching, transplantation and disease association understanding. These are limitations in a circle for advanced clinical practice and research. Conclusion, the ABO blood group system in a concentration of the transfusion medicine and the clinical practice, its genetic basis and clinical significance have two far reaching implications for the healthcare and research. Thank you.